probably pretty helpful for this. I don't know if you guys use the worksheets to study for the first exam, but if you haven't, you might want to because the questions are pretty similar. So we're trying to just define what metabolism is. A lot of our talk is going to be about how enzymes work, because enzymes are what drive metabolism. They're basically what help carry out metabolism. We talk about what the enzyme actually looks like what its structure is. So a lot of this is about enzymes actually, like how they work, what their structure is, how they help metabolism. And then we talk about the different metabolic reactions. So we talk mostly about carbohydrate metabolism because that's our main energy source. But of course you can get energy from proteins and fats. I just don't talk about it quite as much. So if you ever take like a biochemistry class or if you take nutrition, you probably would talk a lot more about it than I do. I spend more emphasis on carbohydrate metabolism. So really metabolism is about energy, acquiring energy. How do you do that? And this will kind of link back to when we discussed the different sources of energy. So we said that like some organisms can use light and some can use chemicals, and then some things can make their own food, and then some things have to eat other food. So we talk about that again a little bit too. All right, so metabolism is the sum of all chemical reactions in a living organism, and it's a balancing act, but it's not like equals in a sense. So what it means is that in order for you to like stay alive, you need to be making more energy than you're burning off. If you reach true equilibrium, that's cell death. Like that's when you die. Like when your energy in is equal to your energy out. That's not what you want. You always want to have a little bit more energy. As far as these chemical reactions, depending on what's happening and what's reacting, you're either releasing energy or you are requiring energy. So a reaction either requires energy to get started or it's going to release some energy that you can use. So this is run by enzymes. Enzymes are what facilitate these chemical reactions. And there's two main types under the umbrella of metabolism, which is catabolism and anabolism, or you can hear it said anabolism. I always just say anabolism though. So do you think you have to compare those? Would you guess? Yeah, right? Like the first part of this class was like comparing some different things, and so this is something that I kind of go over a lot, but people still mix it up at the end, the two of them. So catabolism, the word kata means down. So that's to break things down, and when you break anything apart, that's also called degradation. You're degrading it. So they call them degradative reactions. So when you break something apart, it's going to release energy. And that happens when you break the bonds apart. So it's kind of abstract. You can't like see the energy, but it's in the bonds. You just have to kind of like accept that. And so when you break a bond, that energy gets released. Okay, so those are also called exergonic reactions. Exergonic reactions are where you release more energy than you absorb. And so there's actually ways to calculate that energy, which is not what we have to do, thank God. Like we're not in chemistry class. But if you take chemistry, you would have to like actually do some like calculations related to that. But we're just speaking on it more qualitatively, descriptively. So we're saying that catabolic reactions degrade molecules by breaking the bonds apart. And when you break the bond apart, it releases energy. And then that energy can be used to do work. So work is whatever the cell needs to do to stay alive. It's a bunch of different things. The opposite is anabolism. Ana means up. So this is how you build up molecules. So this is also known as biosynthesis, which means putting together life parts. So you're building a complex molecule from a simple one, and anytime you're trying to build something up, it's going to require energy. So those reactions are going to consume energy. And I think logically that makes sense. Like let's say you think about something that's held together, and let's say it's like really tight. when you break it, you can feel that energy releasing, right? Like you feel the tension kind of release. Now, if you have to put that rope back together, let's say it's a rope, that's going to take energy, right? Because you got to figure out like what the weave pattern is, and you have to like maybe need more pieces of rope to like fix where you ripped it. And so that's going to require energy, and I think it just makes sense logically that if you're trying to build something, it's going to require an energy input for you to do that. So the reactions where you build up are anabolism, and the ones where you break down are catabolism. Cata means down, ana means up. They are opposite reactions to each other, and they're both kind of happening at the same time. So this is visually showing you what happens when you have a glucose molecule. So most of us are probably familiar with glucose because it's kind of like the biological sugar that most living things like. We have to have glucose for our brains to work properly. So if you take in a glucose, you're going to break it down through these processes here, which are glycolysis, Krebs cycle, and oxidative phosphorylation, which is also known as the electron transport chain. These last two steps are what are called respiration. So if you've heard the term cellular respiration, like you know how we breathe in oxygen and breathe out CO2? That happens as part of this process here. So you see the CO2 there, which is the carbon dioxide. And you also end up making a water molecule. And then you lose some energy as heat. And so we're not like 100% efficient. If we were 100% efficient at converting all our food into energy, we'd all be our super sexy, like, meta selves, like perfect. But we don't do it 100% perfectly every time. So some of it gets stored, some of it is lost as heat. Now, when you break that glucose down into these, like, parts, like, you're kind of stripping it away, like, by electrons that are on the atoms. And when you do that, and this is the weird part, because people have asked me, how does this happen? And I'm like, so it's like, it's just a transfer of energy. So the energy that was in the bonds of the glucose gets transferred onto a molecule that's called ATP. So ATP is the usable form of energy. So when you eat food, the energy is kind of dispersed all throughout the food. It's not easy to get to. When you break it apart, though, we can transfer that energy onto a molecule that is easy to use. So ATP, which is adenosine triphosphate, is a high-energy molecule that stores the energy that came from the catabolic reactions. So when you break down food, you store the energy that was in that food as ATP. If you're a eukaryotic organism, you're going to store most of that ATP in the mitochondria, which is what its main purpose is. If you're a plant, you're going to store it in the chloroplast. And if you're bacteria, they don't have organelles, so it's just in the plasma membrane, like that's where their energy is sort of hanging out. So you already have the ingredients to make ATP, because you have the precursor molecule, which is called ADP, and you have phosphorus, like hanging around just naturally. And so you're able to convert it rapidly to ATP, which is the next level, like higher energy. Then if you need to build something back up, so let's say you have enough glucose in your body, and you want to store it as glycogen, if you're an animal, it's going to require an input of energy for you to synthesize a glucose back into a polysaccharide. Because you're taking glucose molecules and you're putting them back together. When you form a bond, it absorbs energy. And so that part of the reaction is going to be this endergonic part, where you actually are absorbing energy to make the reaction. When you first released the energy, that was exergonic. And then you have exergonic on both sides because it's going to release energy from the ATP and transfer it into making that bond. So you can see, hopefully, that it's cyclical, that you basically just can go back and forth between breaking bonds and forming bonds. When you break a bond, it'll release the energy, and then you can use that energy to form new bonds. So it just depends on what the requirements are for the cell. But this is happening all at the same time. So it's not like you're just like, I'm just doing catabolism right now. You're going to be doing it all at once. Okay, any questions on this? It's just a visual representation of catabolism and anabolism. So sometimes, like, I'll show you a reaction, and I'm like, is this catabolic or anabolic? And you just have to look and see what's happening. If I'm breaking bonds apart and releasing energy, then it's catabolic. And if I'm forming bonds and absorbing energy, then it's anabolic. It's not balancing equations or anything. It's literally just like knowing in general what it means to be a catabolic or an anabolic reaction. Another visual representation, I'm saying the same thing, just different ways, different pictures. So catabolism, you take a large molecule and you break it down into smaller parts, and when you do that, it releases energy. So that's an exergonic reaction that releases energy. And that energy is ATP, so it transfers it onto a usable form. With the anabolic reaction, you take smaller parts and you build them back up into a bigger part. That will require an input of energy. And so those are called endergonic reactions because they absorb the energy as part of the reaction. Bonds are being broken. Bonds are being formed. Degradation, biosynthesis. Okay. So, what do you notice the connection is between these two processes? They flip, right? So, if you say that another way, the end material of one could be used as the starting material for the other. And then what's the other connection between them? The other thing that they have in common. The energy, right? So the energy generated from one can be used to fuel the other. And so, like, you kind of literally are what you eat. I like donuts and coffee a lot, but like anything that you eat can be converted into energy to make other things that you need to, like, stay alive. Okay? So this is like a more simplified version of the previous diagram. Okay, so what are these energy sources? Like, where do they come? Where are you getting the energy from your food? Most living things need some kind of carbohydrate, and that's because you have to have a carbon source. To live, everything needs carbon, because we're literally made out of carbon. Like, we're made out of carbon skeletons that are attached to other atoms. You can also use fats, which are lipids. You can also use proteins, which are made out of amino acids. And then, interestingly, nucleic acids you actually make yourself, which kind of makes sense because nucleic acids are what your genetic material is made out of. And it would probably be a problem if you couldn't just make your genetic material. Like, if you had to eat it from food, what if you don't have food? Like, right? Like, what if you're in a starvation mode? It's going to be harder for you to even stay alive if you didn't have the building blocks to make the genetic material. That's just, and this is just how I kind of think of it in my mind. Yes?
So, with the way that right there's put, someone that doesn't eat vegetables literally doesn't need it.
Does it need vegetables?
I don't eat vegetables. None whatsoever.
You need them for other things. Like, vegetables are a source of, I guess, carbohydrates to a point, like starches. But they also have micronutrients, which you could supplement. But they are for the micronutrients and the minerals. So, like, I guess carotene is for eyesight. Like, they've identified the different micronutrients is what they have. But I mean, you can stay alive because you can get it from other stuff.
Like being perfectly healthy, knowing that you haven't eaten vegetables in 20 years.
I don't know.
I'm just saying that's me. I don't eat vegetables at all.
Yeah. I mean,
because based off of that, technically it says that you don't need vegetables for any of that because you need those to live, and literally
you can stay alive without them. Yes. But I don't think that there's a replacement for the way that they come in nature. Does that make sense? Like, so the way that you break it down in nature is different than you would break down like a supplement that had that same stuff, I guess. There are a lot of people who don't eat veggies, like my picky toddler. It drives me crazy. But anyway.
I don't even take a supplement for it. I don't take any type of vitamins or anything.
I guess you are miraculous. I don't know.
Yeah,
I don't really know. You can stay alive eating like hamburgers, like in French fries. Like there's been people that have done that. I mean, I don't know. I can't speak to the health aspect of that. I don't really know. Most people's diet isn't balanced, I guess. Anyway, like naturally, right? Like we're not like perfect little machines where we eat exactly the proportions that they tell us to. There is a reasoning behind why they say to eat certain things, because you're literally made out of the things that you eat, and so you can technically turn the stuff you break your food down into other stuff that you need to make yourself. Okay, so we're going to spend most of our time talking about carbohydrate, how you use carbs. And the other thing that you kind of recognize is that, as we said, the starting material or the ending material of one is the starting material of the other. So technically these arrows or these lines could face in both ways. So for example, you can take a carbohydrate and break it down into a simple sugar, or you can take a simple sugar and build it back into a carbohydrate. So that's what we were talking about with the catabolism and the anabolism. One side is breaking it down, and then the other side is going to build back up. All right, so these are the nutritional modes that I was talking about. And this actually came up sort of when we were talking about the kingdoms and the domains. When I was saying that one of the ways that you could classify organisms by kingdom is based off of their nutritional mode. Like how do they actually use nutrition, like how do they get energy? So autotrophs are things that can make their own food. They're self-feeding. So auto means self or own. So then there's two energy sources. There's either chemicals or there's light, like sunlight. And then the carbon source, if it's CO2, which is carbon dioxide, then that's going to be prokaryotic organisms, and then some things like plants and algae. So if someone was like, okay, what's a plant? You'd say it's a photoautotroph. It makes its own food using sunlight, and its carbon source is carbon dioxide. Well, guess what? Bacteria can do that too. Like that's one of the things bacteria does, and it's one of the things that algae do. So that process of making food with the help of sunlight is photosynthesis. So photosynthetic organisms are the ones that can use sunlight to make their own food. If they don't use sunlight, if they use chemical energy, but they still can make their own food, they call them a chemoautotroph. And really that's just like little bacteria that live in the soil. A lot of the ones that live in the soil are chemoautotroph. So the difference is that they don't use sunlight, they use chemical energy. They use inorganic compounds. And inorganic chemistry tends to be things that like aren't necessarily attached to a carbon, a hydrogen, an oxygen. Like that's what they call like organic. Okay, then you have heterotroph. Heterotrophs are things that have to eat food. They cannot make it themselves. Hetero means other. So they're other feeders or other nourishers. Then you have the two sources of energy again. It's either light or chemical. But in this case, instead of being inorganic compounds, it's organic compounds, like which is a carbon attached to a hydrogen, oxygen, nitrogen. And usually like a carbon skeleton, like a complex carbon skeleton, like a carbohydrate, lipid, protein. So if they do still use light, but they use organic compounds for their carbon source, then those are called a photoheterotroph. So they use light, but they can't make their own food. Like they have to ingest an organic compound. Bacteria, okay, are in that group. So most bacteria are actually chemoheterotroph, but there are photoheterotrophic bacteria, meaning that they require sunlight, but they have to eat food. They're not able to make it themselves. The last one is where we belong. Okay, we as animals are chemoheterotrophs. We don't use sunlight, right? Even though we do use sunlight to synthesize vitamin D, but we don't need it necessarily to like actually make food, like energy, like make energy. But we have to eat, right? Like we have to eat organic compounds, the lipids, the carbohydrates, the fats. And so many bacteria are in this group. Like most bacteria are probably chemoheterotroph. Fungus is in this group. So remember that was another kingdom. And then animals, us, and the worms are in this group also. So one thing to note is that bacteria are in all the groups. So if you were to describe bacteria, you'd be like, they are nutritionally diverse. Like they can be heterotroph, autotroph, chemotroph, phototroph. Like they have a bunch of different, nutritional modes depending on what kind of bacteria it is. Okay. Any questions on this? So the gist of this is that there's two main sources of energy, light or chemical. And then you can either make your food yourself or you cannot. And that's the auto and the hetero part. Bacteria fall into all the categories. Okay, so they're pretty diverse. And I find it helpful if maybe you know an example of each in the category to kind of wrap your head around it. Like most people know that plants are photoautotrophs. They just didn't know that word. But they know that plants use sunlight for their energy. So do you see how this sort of stratifies kingdoms a little bit? Like everyone that's called fungus is going to be a chemoheterotroph. Everyone that's called a plant is going to be a photoautotroph. So that's one of the ways that they also put those groups is by their nutrition, like how they get energy. Okay, catabolism and anabolism, again, is just showing, it's really a bunch of words that I just said already. But I just like to, again, bring your attention to the image that's there. So, catabolism is what's happening down here. Large molecules are broken down into smaller. When you break those bonds apart, that releases energy. And you convert it into a usable form, which is ATP. Then, if you need to build something back up, you can take the parts that you broke down and then reform the bonds with the input of energy, and now you've got the big molecule again. And that side is anabolism. So, one process will generate ATP, and that's catabolism. And the other side will use ATP up, and that's anabolism. All right, so then we've been talking about chemical reactions, and so this is a brief introduction as to what that actually means. What is a chemical reaction? So one of the prevailing theories of what makes a chemical reaction is called collision theory. And it's this idea that as atoms are moving, because they're always moving, they're continually bumping into each other. And if they bump into each other with a certain amount of energy, a bond might be formed or a bond might be broken. So it depends on how frequently they're colliding, and it depends on which molecules are reacting. It kind of depends on the temperature, because the warmer the temperature, the more they'll move around. And so if you think about it, isn't that kind of random? They might bump into each other, right? So there's a way to take out the randomness, and that's what enzymes do. Part of what enzymes do is they make the collisions more likely to happen. They make the collisions more efficient. So they make sure that the molecules come together, and what that does is it actually lowers the activation energy. And that's the minimum amount of energy that has to be there for a reaction to proceed. So if we think about it there's probably a certain amount of force that I have to put on this stand to get it to move. So if I just do this forever, right? It's not going to move, or it's going to move so small that it's going to take 10 years. But if I have enough force behind it, I'm applying enough energy, then I'm going to move it, which is the reaction that I want to have happen. Now let's say that all I'm capable of is doing this. But then one of you is like, let's help her out, right? So then you give me a little push, and then I can actually move it. And that's what the enzymes are kind of doing. They're basically making it so that the reaction will happen more efficiently by physically bringing the reacting things to each other. Otherwise, with collision theory, they'd just be randomly smacking into each other, and sometimes they would react and sometimes they wouldn't. But for a reaction to occur, there's a minimum threshold that has to be met, and that's called the activation energy. And so enzymes lower the activation energy. They make that minimum threshold easier to get over. And we'll say that again, because we'll talk more about enzymes. All right, so enzymes are called a catalyst because they catalyze a reaction, they make it faster, but they themselves are unchanged. So the enzyme does not permanently get altered after the reaction, which is good because that means it can participate in the reaction over and over again. Okay? So the main function of the enzyme is that it speeds up a chemical reaction by lowering the activation energy. So if you look at this diagram here, it pretty much sums that up. And again, since we're not in a chemistry class or biochem, you don't have to make any calculations or anything. Because you could calculate these things, but we're just looking at it more descriptively. So the red reaction is uncatalyzed. Uncatalyzed means without enzyme. And then the blue reaction is catalyzed, and that means with enzyme. So what do you notice the two differences are between those reactions? Just visually. What was that?
The uncatalyzed requires a lot more activation.
Yeah, the uncatalyzed requires a lot more activation energy. And then that translates to what? It's going to take longer. Okay, because if this axis is just time, this time, isn't that longer than that time? Right? So this is literally just showing you what an enzyme does. So the effect of the enzyme is that it's going to lower the activation energy, which is going to speed up the reaction rate, meaning it's going to take less time for you to get to the end that you want. So sometimes I'll call it starting material, because I'm used to kind of talking more, I guess, layman term, but that's also known as reactant. Okay? So the reactants are what is reacting, and then the product is what you're going to make from that reaction. Okay, so enzymes are catalysts. They're not changed during the reaction permanently. Sometimes they'll wiggle a little bit to help the reaction, but they don't actually permanently change. And they lower the activation energy by making the collisions more efficient. And then the reaction is going to take less time if you lower the activation energy. So real life example that I always think of, if you think about getting ready in the morning, and let's say you're not a morning person. So you're all right, some people just get out of bed and get up. But you're I'm that person who's I need to sit here and think about life for 10 minutes. Or hit snooze 5 times. And then I have to have coffee. And then I have to do this, and that, right? So your minimum requirement to get out the door is higher than someone that maybe is naturally able to be a morning person, if that makes sense. So then how could you, as a non-morning person, get out the door more efficiently? You could do all these fabled things like put out your clothes the night before. Decide what you're going to wear and not change your mind like I always do. Decide what you're going to wear. Pack your lunch ahead of time. Get your backpack by the door. If you have kids, it kind of throws a whole new thing in the mix. But a catalyst or an enzyme helper could be a partner that's actually useful, that does things to help you corral the kids so that you can all get out the door. Do you see what I'm saying? So I'm using a more general example of how I view enzymes in my mind. But they're helpers, to help a reaction happen. Because without them, it might not happen. If you're a non-morning person, maybe without your coffee, you're I don't even want to talk to people. So coffee is your catalyst, right? If you have a bunch of crazy little babies running around, Maybe older sibling is a good helper. So they are the catalyst to help you guys all get out the door. So there's different ways you can think about it, but that is what enzymes do. They help with reactions. So what is an enzyme? It's a protein. Most of your genes encode for enzymes. That's how important they are. And they're kind of a globe or globular shaped protein. And they have 6 main classes, which I don't even make you know what those are. If you take nutrition, I think they talk a little bit more about it. But basically, if you see anything that ends in A's, that's an enzyme. And then the other part is that they usually name it after what it does. So like if it's when we talked about bacteria making their cell wall, they have an enzyme that's called transpeptidase. Its job is to make peptide bonds across the cell wall. Trans means across. So usually they'll name it after what it does. You can kind of guess what it does. So this enzyme, the purple thing, is the enzyme. It's protein. It's called sucrase. So its job is to break down sucrose. So if you see something with ose, that means sugar. So sucrose is a sugar. It's a disaccharide. It's made out of 2 little bits. And so what the enzyme will do is that it has a special pocket in there that's called the active site. And that's where the chemical reaction is actually going to occur. So what do you notice about the shape of the active site? If you look at what's happening here. It has to match the thing that's trying to react, right? And that's how they make the enzyme specific, because not just anything can sit in the active site. It has to be the right size and the right chemistry, I guess, too. So the active site of the enzyme, the shape of it is super important. And if that shape gets altered for some reason, which we'll talk about, the enzyme might not work anymore. So the active site is where the reaction occurs. It's where the reactant, which is also known as the substrate. So there's more than one way to say it. Starting material, reactant, or substrate all mean the same thing. Okay? So the substrate sits in the active site. And then what happens to that substrate at the end? It broke apart. So what kind of enzyme is this? Degradation. So it's a catabolic reaction, right? So it's a degradative reaction. And notice that the bond was split apart with the addition of a water. And that's why water is so important, because you literally need it for your metabolism. It participates in the reactions. So did the enzyme change during the process? The big purple thing? No, it looks the same, right? So that means the reaction could happen again. So looking at this diagram, what things would change the reaction rate? What would slow it down? Based off of the components that you're seeing that need to be there.
Not having water.
Not having water could slow it down, right? For sure. What else could slow it down? What do you need to start with? You need the sugar, right? You need the substrate. So if you don't have the substrate, that's going to slow down the reaction. What if something bad happened to that enzyme? What if I karate chopped it into pieces? Is it going to work? No, right? Because then I might have changed the shape of the active site. So I think by thinking about those things too, you can think about how enzymes work. What if I blocked the active site? What would happen to the reaction? It wouldn't occur. And that's when we start talking about inhibitors. What could speed up this reaction? Make it go faster or at its maximum rate. So what are the components? We said we need an enzyme and a substrate, right? So will it speed up if you have more substrate? Yeah, because you have more substrate, right? So you're going to have more reaction. I guess technically if you had more enzyme, it would do that. If the heat was increased a little bit, maybe it would. Like, it's going to happen faster in warm conditions probably than cold. But eventually, all the enzymes would become saturated. Like, so they can only go as fast as there's active site available, if that makes sense. Like, eventually, like, the sucrose won't have anywhere to, like, bond to. So the reaction would, like, slow down. What if you were just like, I don't need sugar right now? What do you think would happen to the reaction? If you received a message saying, I don't need sugar right now, I don't need to break this down, I have enough. Do you think the reaction is going to keep going? No, it's going to slow down, right? Because like it doesn't make sense to keep making a product that you don't need. So then you could store the excess, and then you would receive a signal later that says, okay, we're actually low again, so let's turn this back on. That's called feedback inhibition. It's like a feedback mechanism. And a lot of it's like genetically, like there's genetic regulation of this. All right, let me click on this video. I don't remember which one this one is. There's some that I like better than others. I can't remember if this is one that I like or not. Oh yeah, this one's short. Okay.
Is it odd to have a favorite protein? Well, I don't think so. Probably because my favorite protein happens to remind me of one of my favorite childhood games, Pac-Man. If you haven't played Pac-Man before, then chances are you're much older than you. But now you can play it on Google. Just Google Pac-Man. It's a Google Doodle. Anyway, I digress. But see, in Pac-Man, you have this little character. It goes around, finds these pebbles, and the pebbles fit right into it. Well, a lot of illustrations that you will find of enzymes, they happen to look, at least us, a lot like Pac-Man. I remember P for Pac-Man and P for protein. Most enzymes are proteins. Now, in the game, we mentioned there are these little pebbles that Pac-Man goes after. Well, enzymes have a specifically shaped area called an active site, where items can bind. The items are called substrates. It's very specific binding because the active site is specifically shaped for the substrate that binds there. Very specific. So what happens when a substrate binds an enzyme? Well, usually the substrate is held there with some weak bonds because it's not going to stay there forever. Something called induced fit will happen, which means the active site can change its shape even more to bind that substrate, like an enzyme substrate hug. The enzyme can either build up or break down the substrates that specifically bind to it, and we call the resulting item the product. An enzyme has the ability to really speed up reactions. Reactions that technically could happen on their own, but with the help of enzymes, they can be sped up to make processes effective for life. Let me give you a great real-life example. The enzyme lactase. Another really cool thing about enzymes is that they often end in ASE, like lactase. Many sugars, on the other hand, end in OSE, and lactose is an example of a sugar. Lactose is a disaccharide, meaning it contains two sugar molecules bound together. We don't actually digest it so well in that form. It's big. The enzyme lactase has the ability to break lactose down into smaller parts that our body can digest. And this is a lot better option than waiting for a chemical reaction with lactose to happen spontaneously. Now, with the lactase enzyme, lactose can be broken down quickly and digested. Now, there are some people that do not produce enough lactase. They can be what we call lactose intolerant, which means that if they consume foods that have lactose, such as milk, it can make them sick. They can't break the lactose down efficiently without lactase. In that example, one thing to point out, lactase, the enzyme, can break down a lot of lactose, the substrate. The lactase doesn't get used up in the reaction. It's still there. We call enzymes a catalyst because they can be used over and over and over and over in the reaction. By the way, your digestive system uses all kinds of enzymes. You have lipase, and that breaks down lipids, which are fats. You have amylase, which breaks down starch. You have protease, which breaks down appreciate it, but essential little helpers are called cofactors and coenzymes. They may bind to the substrate or to the active site. They help the enzyme do its job of building up or breaking down substrates into products. Now, you didn't forget our Pac-Man analogy yet, right? In the game Pac-Man, there are these ghosts, and when they touch the Pac-Man, it makes this sound. It's like, and the Pac-Man shape gets all distorted in the process. So what does this have to do with enzymes? No, there aren't ghosts around, but enzymes do have certain ideal conditions that they like. For example, an enzyme that is in your stomach would have an ideal pH that is very acidic because the environment in your stomach is very acidic. Different enzymes have different ideal pH and temperature ranges. If an environment changes out of an enzyme's ideal pH or temperature range, then something that reminds me a lot of that horrible sound I tried to make can happen. The enzyme becomes denatured. That means its shape becomes distorted. It can no longer bind to its substrate. It can no longer work correctly. It is finished. Well, that's a dramatic end to enzymes. Keep in mind that if you have an interest in this topic, many medical researchers have a large focus on enzymes. Enzymes regulate a lot of the body processes, and many diseases can involve specific enzyme production, or the lack of it. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so that was a good little summary of a lot of what we just talked about and some of the things that are going to come up here in a minute. Okay, so they mentioned in the video about denaturation, which is where the enzyme changes shape, and it can be permanent or not. It just depends on how bad the assault was. So the active site has to be the correct shape in order for the substrate to bind, but sometimes it can kind of wiggle a little bit to make an even tighter fit, and that's the induced fit that they were talking about. So I think of it as like, if you think of enzyme and substrate as a lock and key mechanism, then the enzyme is going to be the lock and the substrate is going to be the key. And you know how sometimes you have a lock where it'll only work if you like jiggle it a little, or like I had one that I had to pull the handle to like get the key to like turn. And so it's the idea that you can get an even better fit if you adjust a little bit, but it's not a permanent change. So that's why they're saying the enzymes don't permanently change. So one of the things I usually ask you is like, if you saw a simple diagram that had an enzymatic reaction, that you could say if it was catabolic or anabolic. So the synthesis building reaction is at the top. The enzyme has the right shape for those substrates, and it brings them together, and then it builds something back up. Then a catabolic enzyme, it takes a bigger thing and it breaks it back down. But in both cases, the enzyme did not change shape permanently, and the active site was intact so that the substrate could actually fit into the active site. All right, so they mentioned in the video the coenzymes and the cofactors. So cofactors are typically minerals or metal ions that help form a bridge between the substrate and the enzyme so that they can stay together. And then coenzymes are typically vitamins, and you don't have to know which vitamins do which. There's a table in the book that references it. If you've had nutrition, you might remember which vitamins do what. But they're important for the enzyme to actually be active. So the enzyme is not always in its active form. And if you were low on certain micronutrients like minerals and vitamins, then some of your enzymes might not work as effectively because those things sit in the active site, which is also known as the catalytic site, so that the reaction can occur more efficiently. So when it's fully formed with the enzyme and the cofactor and the coenzyme, they just call it a holoenzyme. So one thing to note is that there are some enzymes that are important to life, and they tend to be conserved across life. So an example of that is catalase. Catalase is an important enzyme that helps break down reactive oxygen compounds to protect your cells from damage. And so if you compare yeast to human catalase, chemically they look like super similar. Like they're not exactly the same, but their protein structure is like pretty close. What do you remember about yeast and humans? What do they have in common? What's the cell type? eukaryotic, right? So they have that in common. And so in that domain of eukaryotic organisms, a lot of things have catalase. But you know what? Bacteria have catalase too. So what I would conclude about catalase is that, wow, it must be important for life, right? Because so many different living things have catalase enzyme. Okay. All right, so this is just going back to what the enzyme does again. So I mentioned that there's a lock and key mechanism. Lock is the enzyme, and the key would be the substrate. But sometimes they can flex a little bit together, and that's called induced fit, so that'll get you a better connection. They mentioned the enzyme lead in the little video. It's not like one enzyme is working by itself. They're usually working in a metabolic pathway that's super complicated. And even in here, like when we go through glycolysis, there's like 6 enzymes in the first step. I don't make you know any of those enzymes, but if you ever had to take biochemistry, you might have to know like some of those enzymes. So a compound can be a substrate for several different enzymes. So what it turns into depends on the enzyme that was acting on it. Because all enzymes can only catalyze their reaction that they do. So let's say you had enzymes 1, 2, and 3, and you had a substrate that was a circle. Well, enzyme number 1 might be able to turn the circle into a triangle. Enzyme number 2 can turn the circle into a square. Enzyme number 3 can turn that same circle into like a pentagon. So they're different enzymes, they catalyze different reactions. So even though it's the same substrate, what gets made is different. And so a lot of times what happens in a metabolic pathway is that the substrate for one, in that enzyme, the product that is generated will be the substrate for the next enzyme. And so it kind of carries on in this like little chain. Okay, so this is showing the lock and key model, and it's what it sounds like. Okay, so the enzyme would be the lock, and the substrate would be the key. They will temporarily come together and make that enzyme substrate complex, and then the reaction will get catalyzed with the help of cofactors and coenzymes that are sitting in the active site, and then that product, whatever it is, will get released. So then all induced fit is showing is that there's a conformational change that occurs to make that binding even tighter. So when the substrate first comes in, it might be kind of like this, and then when they go induced fit, it'll go like this. Okay, so it'll just make like a tighter bonding so that you're more likely to catalyze that reaction even better. All right, so they mentioned that proteins, like enzymes, can get denatured. Denaturing is where it loses its three-dimensional shape. So enzymes have levels to their shape. There's primary, secondary, tertiary, and quaternary. The quaternary structure is the final three-dimensional shape that it takes on. And that shape is important because if you change a lock, then the key won't work, right? And that's essentially what's happening with the denaturing, is that you're potentially impacting the active site so that the substrate can't fit in there. The two things that do that most commonly are excessive heat and excessive pH. So they mentioned that most enzymes have an ideal condition that they work the best at, where they can catalyze the reaction the fastest. Any deviations in that can be catastrophic to the function of the enzyme. So think about human blood. I mean, we have a very narrow range of pH that is acceptable before we start having problems. If the blood gets too acidic or too basic, then we're not able to sustain life properly under those conditions. And a lot of it probably has to do with proteins that are important are getting denatured. So a real life example of this, if anyone uses, say, a heat product, a heat tool to straighten your hair, so you use a blow dryer or a flat iron or whatever. So if you have curly hair, the proteins in your hair are actually coiled. When you apply heat, it temporarily breaks the bonds that are holding the coil, and the hair will go straight. If you add water back, it reforms those bonds. That's why it's if you get a blowout or your hair straightened, this is the worst thing that can happen, right? When you walk out. So it'll curl back up with the addition of water. Now, if you overdo it with the heat tools, like if you're using the temperature too high or you're doing it too frequently, you'll notice eventually the curls will not go back to normal. Like you'll have some of them that are like damaged. And so you can permanently denature that hair. It'll still grow curly at the roots, but that hair has become damaged and it might not take back that spiral shape after a while. So using heat tools would be a potentially reversible denaturation, because as soon as you add water, it's going to technically reverse. Now, a permanent denaturation of your hair would be if you used like an at-home, like the straightening perms. Like they're basically made out of lye, and lye is a strong base, and it will permanently break the bonds in the curls. And so then all the hair that is out of your root will be straight. But of course you didn't alter yourself genetically, so the new hair is going to grow back in curly. But when you add that chemical straightener is going to permanently denature those proteins. So this is an example where it's not a matter of life and death, right? Like it's just your hair. But that's the impact of excessive pH, excessive heat, is that you are going to denature the protein. It's going to lose its three-dimensional shape. And if it's an enzyme, then that means the enzyme probably isn't going to work the way it's supposed to. So this was the hair example that I gave you guys. If you apply heat, and really everyone's hair is different because the structure of hair is different. Some hair is more heat resistant. Like my hair will not straighten from a blow dryer. Like I have to use a flat iron. Like it'll just make it really big and not straight. So, it depends on your hair, like how it takes the heat and how thick the hair is. But this is just showing that under electron microscope, you can see where the hair is like kind of peeling, like because it's like these layers of keratin that make up hair, like, and then there's the cuticle, which is the outside of the hair. And so if you overdo it with the heat, it can start lifting up the cuticle, and that's when you start to get like frizziness. And that can happen with other damage too, like pulling on the hair too much, or dyeing it too much, like bleaching it too much, too many times in a row, trying to fix things, stuff like that. Okay. All right. The other thing, which I actually find these really interesting, are the inhibitors. So they mentioned that enzymes can be inhibited in different ways. So a competitive inhibitor is where it competes for the active site. So those are molecules that will actually sit in the active site, and if the active site is occupied, the substrate can't get in. So I always think about like when we were kids, there was like 5 of us, and we always lived in like small little apartments. And it's like if someone like stole your seat, then you're like, I don't have anywhere to sit. And then if it was my brother, I'm like, I'm sitting on you, and I would just try to sit on him, but I still couldn't get in the seat, right? Because he was in the way. And so that's what happens with these competitive inhibitors. They compete for that space. So the substrate cannot get in, and so the reaction will not occur. Now the other type of inhibition is called allosteric. It's not the active site that's getting inhibited, but the inhibitor is binding to a allosteric site, which is a non-competitive site, and it changes the shape of the active site. So the active site was this shape, and now with this non-competitive inhibitor, it becomes this shape. The reaction would not occur. Okay. So let's say I was trying to sit in a chair, but I took off one of the legs, right? And I'm like, I can't really sit in it anymore. But if the seat is the active site, like the actual seat, I didn't really change that necessarily, but I did change the overall composition of the chair. So the outcome is that I can't sit down. Okay. So the reason that knowing about inhibitors is kind of cool is because a lot of medications that are used are enzyme inhibitors of some sort. Okay, so here's some examples of inhibitors. And it's not that you have to remember exactly how these all work. This is just illustrating the point. Okay. So the first one are penicillin drugs, which I've actually mentioned this before. Penicillin drugs interfere with the cell wall of bacteria. So there's an enzyme that bacteria have that's called a transpeptidase. The job of this enzyme is to go through and make the cross links in the peptidoglycan wall. And so if you take a drug like penicillin, penicillin is a competitive inhibitor of this transpeptidase. So it will compete for the active site because it can bind the active site too, but it binds permanently. So this is an irreversible competitive inhibitor. And that's why the bacteria die. Because if you bind up this active site, the cell wall is not going to get made properly. So if you imagine that these links are gone, the whole thing's just going to like fall apart. So that's how penicillin drug, any drug that's an illin, or if it's called a beta-lactamase, those are drugs that work in this way. There are irreversible competitive inhibitors of the bacterial transpeptidase. The outcome is that the cell wall doesn't get made. Okay, and what I just said was the mechanism of action, that it's a competitive inhibitor. Okay, this example, I was like, I watched too many murder shows. This is showing what happens with cyanide, or carbon monoxide acts the same way. So this is an example of a non-competitive inhibitor. So in the electron transport chain, which is how you make most of your energy, it's a series of proteins that act in a row, like in succession, to move these electrons through. And it ends up pumping hydrogens outside. And then there's a separation in charge across the membrane. That forces the hydrogens back inside this enzyme over here that's called ATP synthase. And you end up making a bunch of ATP at one time. Well, what carbon monoxide will do, or cyanide, is that it will bind to the last little protein in this chain, and that shuts it down so that that final step doesn't occur, and you end up having cell death very quickly because you're not able to make ATP. So you shut down the electron transport chain, and the electrons can't be released to the oxygen because the oxygen is what is waiting here at the end to catch the electrons so that you can make that energy. So this process is getting blocked. It's not binding to the active site. It's binding to another site, but that's altering the function of the active site. I think carbon monoxide, too, actually binds to hemoglobin more tightly than oxygen does, and that's another problem with carbon monoxide poisoning. But it also does this to the electron transport chain, which is really bad for you. Like the cell's going to die from that. Okay, this is showing just the change in the rate of reaction. A normal enzyme would be in purple. And then if you have a competitive inhibitor, you're going to see a lower reaction. Because I think sometimes competitive inhibitors can be overcome. Like you might be able to, it might not be irreversible. Like I guess it can actually pop out of the active site. So you still retain some rate of reaction. And then non-competitive inhibitors actually change the shape of the active site. So those really slow down the rate of the reaction. But notice it's not to zero, right? Because especially if you think about medications, there's probably very few medications that you would want the enzyme to never work. You just want to slow it down so that you can manipulate whatever process you're trying to manipulate. So for example, if you think about ACE inhibitors, the angiotensin converting enzyme inhibitors, their job is to relax the blood vessels so that you lower the blood pressure. But you don't want it to be totally relaxed where you're not having any flow. Does that make sense? So it's going to shut it down, but not totally. Okay. All right. The last example of inhibition is called feedback inhibition. So this is the one where I was saying that if you have enough of the end product, you don't need to make more of it. So typically things will act in a pathway, a collection of enzymes. This is a very oversimplified example because it could be dozens of enzymes that are reacting with each other. So let's say the job of enzyme 1 is to turn green triangle into purple rectangle. And then you take purple rectangle and turn it into blue squiggly thing. And then blue squiggly thing gets turned into square, yellow square. And yellow square is what you want. Okay, whatever it is, sugar or whatever. So if you have enough yellow square, then it's like, all right, it's not energetically favorable for me to keep making more yellow square. I already have enough yellow square. So there'll be a feedback mechanism where if there's an excess amount of yellow square, which is end product, it'll feed back on the enzyme that started the process. And when it's there, it changes the shape of the active site. So in its active form, this enzyme has a triangular active site. In its inactive form, it's more like this square shape. Then what will happen is, let's say you get low on yellow square again. That'll trigger this to come off. And then it'll revert back to the active form, and then you'll start making yellow square again. So this is something that your body just does to regulate everything, I guess, like, so that you don't waste energy trying to make things that you don't need anymore.
Feedback.
What was that?
Feedback.
Feedback inhibition. That is what this is called. So this is an example of feedback inhibition in bacteria, the way they regulate their amino acids. So basically, this one's like a little bit complicated, but typically the way bacteria operate their genes is a set of genes that have a similar function will be operating together in what's called an operon. So the genes are actually like literally right next to each other on the chromosome. So there's like these 5 genes that are involved in making tryptophan, which is one of the essential amino acids. And so tryptophan is actually, the presence of it is what can slow this process down. So it's an example of feedback inhibition because if tryptophan is present, then it's going to activate the repressor, which is going to repress this process. Okay, so it's a little bit convoluted. But basically, it's the tryptophan, which is the end product that you want, that will activate the repressor, which will stop you from making tryptophan. Okay, so it's still a type of feedback inhibition. And again, the regulation of this is genetic. The genes that regulate this tryptophan process work together, and they're by each other. And a lot of times you'll see in bacteria especially that genes that have a related function will just be next to each other, like on the chromosome, and then those proteins will work together as part of this enzymatic process. Okay. All right. I'm actually going to stop there for now because we're kind of switching gears to like a different thing. So I'll pick up with this next time. We'll take a little break. We'll start at 25. So 11:25, we'll start lab. Okay? All right. I'll see you guys next time. Be safe out there.